Hello everyone and welcome. Thank you for joining this webinar. Every year, hundreds of scientific papers describe exciting new stem cell-derived models, yet only a handful ever become technologies that pharmaceutical companies routinely use and regulators are willing to consider as part of drug development. Why? That's the question I'd like to explore today. The answer isn't simply better biology, it's predictivity, reproducibility, standardization, and demonstrating that a model is fit for its intended purpose. Ultimately, the value of a stem cell-derived model isn't determined by how closely it resembles human tissue in the laboratory. It's determined by whether researchers can generate reproducible data across different laboratories that are robust enough to support important scientific and regulatory decisions. I think that's one of the defining challenges and opportunities for the stem cell field today. The next decade of stem cell biology won't be defined simply by creating increasingly complex cell models. It'll be defined by making those models reproducible, functionally characterized, and trusted enough to support real-world decisions. Fortunately, we're beginning to see exactly how that transition can happen. Over the past decade, a small number of human stem cell-derived assays have evolved from promising research tools into technologies that are influencing drug discovery and safety assessment. They're helping pharmaceutical companies make better decisions earlier in development and increasingly providing evidence that regulators are willing to consider. In this webinar, I'd like to explore how that transformation happens. To explore that question, I'd like to share two examples that I've been fortunate to work on over the past decade. Together, they illustrate how stem cell-derived assays evolve from promising biological models into trusted NAMs that support drug development decisions. The first is the human iPSC cardiomyocyte multi-electrode array assay for assessing proarrhythmic risk. This is one of the most mature examples of a stem cell-derived NAM, progressing from academic research into regulatory application. The second is the emerging human iPSC neural MEA assay for seizure liability assessment. Although this field is at an earlier stage, we're beginning to see many of the same ingredients that made the cardiac assay successful: international collaboration, assay standardization, functional benchmarking, and growing regulatory engagement. Together, these two case studies illustrate how stem cell-derived NAMs mature from exciting biological innovations into reproducible, standardized assays that can have a real impact on drug development. Before we dive into those examples, it's worth briefly defining what we mean by a new approach methodology, or NAM. NAMs are technologies or approaches that provide information for hazard and risk assessment while reducing reliance on vertebrate animal testing. Importantly, the goal isn't just to simply replace animals. The goal is to improve the prediction of human biology. Despite decades of progress, late-stage failures in drug development remain common, and many ultimately reflect limitations in how well our preclinical models translate to patients. At the same time, advances in stem cell biology, electrophysiology, and computational analysis have dramatically improved our ability to model human physiology in vitro. That convergence has created a genuine inflection point for human stem cell-derived models, making them increasingly capable of supporting real decisions in drug discovery and safety assessment. It's important to recognize that this shift didn't happen overnight. Regulatory engagement with NAMs has been building for more than a decade through initiatives such as CiPA and other FDA-led validation efforts. The FDA Modernization Act 2.0 didn't start this transition. It recognized that the science had matured. It gave regulators flexibility to consider the most scientifically appropriate evidence while maintaining the same safety standards. So what does a successful NAM actually look like in practice? Here is an example of a NAM that's already crossed the line from experimental to operational. Cardiac safety assessment, an area where the cost of getting it wrong is high and where regulators have been deeply involved in defining what better prediction should look like. Since 1989, 14 drugs have been withdrawn worldwide due to torsades de pointes, TDP, a rare but potentially fatal ventricular arrhythmia that can lead to sudden cardiac death. It's characterized by a rapid heart rhythm originating from the ventricles, QRS complexes that change shape and appear to twist around the ECG baseline, and an underlying long QT interval. To mitigate risk of developing potentially pro-arrhythmic drugs, the International Council of Harmonization, ICH, issued two guidelines in 2005, S7B and E14. E14 is the ICH clinical guideline that explains how to evaluate whether a drug prolongs the QT-QTC interval and increases pro-arrhythmic risk in humans. Its counterpart, S7B, is a non-clinical safety pharmacology guideline. In plain terms, it tells drug developers how to assess whether a new drug might delay cardiac repolarization and prolong the QT interval, which can increase the risk of dangerous heart rhythm disturbances such as TDP. S7B's objective is to identify a drug's potential to cause delayed ventricular repolarization early in development using preclinical studies so that clinical risk can be better managed. S7B recommended evaluating the risk through two tests. One, an in vitro assay of IKR hERG potassium channel block. IKR inhibition is strongly associated with QT prolongation. Two, an in vitro QT prolongation study in animals, typically in beagle dogs. These measures have reduced the likelihood of drugs with hidden TTP liability reaching the market. However, these tests focus on surrogates, QT prolongation and hERG block, not the mechanism that actually causes arrhythmia, and so there are concerns that overemphasis on these endpoints may have hindered safe, effective drugs reaching the market. Human-induced pluripotent stem cell-derived cardiomyocytes have been proposed as a better model for predicting proarrhythmia because they capture human-relevant integrated cardiac electrophysiology in ways that traditional assays cannot. The argument is not that they replace the established models, but they address key blind spots in the older approaches. Human iPSC-derived cardiomyocytes express multiple interacting ion channels simultaneously and possess many of the structural and functional aspects of cardiomyocyte biology, thereby providing a system-based model for a system-based problem. Because arrhythmias arise from disturbed electrical conduction in the heart, Axion's Maestro multi-well MEA system is an ideal platform to pair with human iPSC-derived cardiomyocytes for assessing proarrhythmic risk. In the human iPSC cardiomyocyte MEA proarrhythmia assay, the cardiomyocytes are cultured on plates containing microelectrodes, allowing non-invasive recording of electrical activity following drug exposure. The assay measures changes in field potential duration, FPD, a QT-like surrogate, beat rate, conduction, and rhythm. Together, these measurements provide a direct functional readout of cardiomyocyte electrophysiology and key endpoints relevant to proarrhythmia risk. Of course, none of this progress happened by chance. It required an unprecedented level of collaboration between regulators, industry, and academia. The FDA-led CiPA initiative, the Comprehensive In Vitro Proarrhythmia Assay Initiative, was launched in 2013, specifically to improve prediction of drug-induced TDP risk beyond the traditional hERG-QT paradigm. CiPA brought together regulators, industry, and academic groups in a coordinated international effort. CiPA proposed a mechanistic, integrated framework with 4 pillars: multi-ion channel pharmacology with human cardiac ion channels, in silico human ventricular models, early clinical ECG analysis, and human iPSC-derived cardiomyocytes to verify integrated cellular effects. The human iPSC-derived cardiomyocyte arm was designed as a biological reality check to confirm whether predicted risks manifest as arrhythmia-like behavior in human cells for the in vitro cardiomyocyte pillar. It is therefore unsurprising that the MEA assay was selected as the platform of choice, as it offers a direct and integrated readout of cardiomyocyte electrophysiology. 2 multi-center CiPA studies involving pharmaceutical companies such as BMS, Eisai, Genentech and J&J confirmed the human iPSC cardiomyocyte MEA assay can detect delayed repolarization and arrhythmia-like events linked to TDP risk. These CiPA findings directly informed the ICH E14 S7b question and answers that were published in 2022, which recognized human iPSC-derived cardiomyocyte data as supportive evidence for proarrhythmic risk evaluation. But perhaps even more important is what happened afterwards. A recent FDA analysis reviewed IND submissions between 2012 and 2025 that included human iPSC cardiomyocyte data. An investigational new drug, IND, is a regulatory application submitted to the US FDA that allows a sponsor to begin clinical trials in humans with a new drug or biologic that has not yet been approved. 48 IND submissions have been submitted to the FDA CDER with human iPSC-derived cardiomyocyte MEA impedance studies as supporting data. These human iPSC cardiomyocyte studies demonstrated higher overall accuracy in predicting clinical QT prolongation risk compared to standard hERG and in vivo non-rodent QT studies alone. Notably, to achieve comparable accuracy to human iPSC cardiomyocytes, additional animal studies beyond the standard in vivo QT assessment were needed. Interestingly, the authors from the FDA went on to propose several scenarios where human iPSC cardiomyocyte studies could be considered as a replacement for in vivo non-rodent QT studies, shown here in this table. Perhaps one of the clearest indicators that a technology has matured is when it becomes routine. Today, more than a dozen CROs offer CiPA-style human iPSC cardiomyocyte MEA assays using Axion's Maestro platform as standard services. The FDA now offers a qualification pathway for NAMs. In 2020, the FDA launched the Innovative Science and Technology Approaches for New Drugs ISTAND program. This is an FDA program that supports the development of novel drug development tools such as microphysiological systems, also known as organ-on-a-chip, artificial intelligence-based algorithms, and other innovative technologies not covered by existing qualification programs. The program's goal is to qualify these new technologies to facilitate drug development and streamline the review process, ultimately helping to bring safe and effective new medications to patients faster. Axion submitted a letter of intent to the FDA in September 2025, seeking acceptance of the human iPSC cardiomyocyte MEA assay into the ISTAND NAMs qualification pathway. I'm pleased to say that the submission was accepted in December. It became the 14th project accepted into ISTAND and the first to utilize a stem cell-derived model. That represents a remarkable transition. In just over a decade, the assay evolved from an emerging academic model into a regulatory-facing NAM with broad industry adoption. Looking back at the evolution of the cardiac assay and now watching similar efforts emerge in neural safety, one lesson really stands out to me. Standardization is rarely the most exciting part of science, but it's often the step that determines whether a technology is widely adopted. Developing human-relevant biology is essential, but biology alone isn't enough. Researchers, pharmaceutical companies, CROs, and regulators all need confidence that an assay will generate reproducible, interpretable data, regardless of where it's performed. That's why I think the cardiac story is so important. It doesn't simply show that human stem cell-derived cardiomyocytes work, it shows how a promising biological innovation becomes a reproducible standardized assay that can generate evidence to support drug development decisions and regulatory evaluation. That naturally raises the next question. Can we apply the same roadmap to other stem cell-derived NAMs? One of the best places to explore that question is neural safety. In many ways, the neural field today resembles where cardiac safety was roughly 5 years ago. The biology is compelling, industry interest is growing, and the challenge is now how to build the evidence and standardization needed for broader adoption. Seizure liability represents a very different biological and translational challenge. Drug-induced seizure risk remains a major cause of compound attrition and post-market safety concern. Seizure is an in vivo event that refers to a period of rhythmic, synchronized, abnormal neural activity that may result in tonic-clonic convulsion and/or more subtle effects such as visual disturbances, tingling, or mood changes. Yet seizure liability remains difficult to assess preclinically. Current approaches rely heavily on behavioral observations in rodents, and behavioral observations can be difficult to interpret and often lack sensitivity at pharmacologically relevant doses. This is why MEA technology became attractive for seizure liability assessment. The MEA assay directly records spontaneous neuronal network activity and network synchronization. Historically, primary rat cortical neurons were the dominant model because they form highly reproducible excitatory inhibitory networks. But over the last several years, there's been growing interest in moving towards human iPSC-derived neural systems. The motivation is obvious. The CNS translational gap remains substantial. Species differences in ion channel expression, receptor pharmacology, and seizure thresholds all complicate interpretation of animal data. Human neural cultures offer the potential for improved mechanistic relevance. But unlike cardiac safety, neural MEA assays have suffered from a lack of standardization. Different laboratories use different cell models, media formulations, maturation times, analysis parameters, and activity metrics, and that variability has limited broader adoption. To move the field forward, we need greater consensus on what constitutes a functionally mature and reproducible neural assay. To address this challenge, we brought together leading experts from across academia, industry, and CROs to establish a community-driven functional framework for the human iPSC neural seizure liability MEA assay. This collaborative effort involves scientists from 5 pharmaceutical companies, 3 CROs, 4 academic groups, and Axion BioSystems. Together, we reviewed evidence from 11 published studies alongside the practical experience of 11 laboratories that have been using this assay for many years. Our goal was straightforward: to define practical functional acceptance criteria and reporting standards that improve reproducibility, enable meaningful comparison across studies and laboratories, and ultimately support broader adoption of the assay. One important conclusion from this work is that the spontaneous baseline neural activity matters a lot. To reliably detect seizogenic compounds, the neural network must first exhibit sufficiently mature and coordinated activity. So we proposed quantitative baseline acceptance criteria, including minimum active electrodes, minimum network burst participation, and network burst frequency. We also proposed using a standard positive control, picrotoxin. And several recent studies have demonstrated encouraging translational potential. For example, work from ASIS showed that changes in network burst frequency in the MEA assay could predict seizure-associated cerebral spinal fluid concentrations for certain compounds in vivo. And BMS recently described how an integrated MEA workflow could have helped identify unexpected seizure liability in a development compound before more extensive downstream studies. These examples matter because they illustrate something important. The assay is moving beyond academic curiosity towards practical decision support. Now, I want to be careful not to overstate where the field is today. The human iPSC neural seizure liability MEA assay is not yet where the cardiomyocyte assay is in terms of maturity, but the trajectory is becoming familiar. In other words, we're beginning to see the same roadmap emerge that transformed the cardiac assay into a regulatory-facing technology: standardization, reproducibility, collaboration, shared data sets. In many ways, the neural field today resembles where cardiac safety was approximately 5 years ago. So what broader lessons can we take from these examples? I think there are 3. First, the most successful NAMs are not trying to recreate the entire human body. They're designed with a specific context of use in mind. The goal is not maximum biological complexity. The goal is sufficient biological relevance to answer a specific question better than existing approaches. Second, standardization matters enormously. Without shared functional benchmarks and reproducibility frameworks, adoption stalls. CiPA demonstrated that coordinated validation efforts can fundamentally change confidence in a technology, and the neural field is now beginning a similar process. And finally, the value of stem cell-derived NAMs is not just reduction of animal testing. The value is earlier, more human-relevant decision-making. Fewer false positives, fewer late-stage surprises, and ultimately better prioritization of compounds before expensive downstream studies. That is where these technologies can have a real-world impact. So let's return to the question I asked at the beginning of this webinar. Why do only a handful of stem cell models become assays that industry adopts and regulators are willing to consider as part of drug development? It's not because they're the most complex. It's because they're reproducible, they're functionally characterized, they're standardized, and they generate data that scientists, pharmaceutical companies, and regulators can interpret with confidence. Ultimately, I believe the future of stem cell-derived NAMs won't be determined by how sophisticated our models become. It'll be determined by how well those models generate reliable, human-relevant evidence that can support better drug development decisions. Thank you very much.